Everyone wants to treat cancer better. And perhaps one of the most popular methods today is by using the body's own immune cells, cell-based immunotherapies. However, the failure rate in clinical trials is over 96%, and it costs over $500 million to develop an approved drug. We have a way to improve that. One of the most important parts of the T cell of the body is the T cell. These bind to cancer cells and kill them. But if they bind too weakly, they fail. And if they bind too strongly, they cannot move on to the next tumor cell. Our technology picks T cells with just the right amount of avidity for their targets. This is done by binding a large population of T cells to their targets and gradually increasing the force we try to put them with. By using microfluidics, this can be done on a benchtop device. This technology is a better predictor of clinical outcomes compared to traditional in vitro assays. It has the potential to reduce the number of tests, failed animal studies, and unsuccessful phase 1 clinical trials. Compared to our nearest competitor, which can only handle 400 T cells at a time, ours can handle over 10,000. It has the unique ability to recover all the cells post-analysis for further evaluation. Our prototype works brilliantly. However, it requires manual intervention. Technicians must painstakingly pipette out the cells each time they use the chip. To automate this process, we are seeking an investment of £500,000. The result will be a pre-production version of the device that we believe will be ready to test with pharmaceutical companies that want to make their cancer immunotherapies safer and more effective. My name is Marwin, and please join me if you want to change the world one cell at a time. Thank you.
So this brings us to the final 2 questions from our wonderful scrutinizers today. Marwin, I'm sure you're ready. Oh, there you go. You were perfectly timed.
Just describe, what's unique about your intellectual property position around your screening technology.
So we have filed for international IP, and it's going through the national phases right now. It was so the part is using the shear stress in a microfluidic device, and that has not been done and for this specific purpose.
Did you say shear test?
Shear stress.
Shear stress.
My apologies.
It is an engineering department, after all.
It's in micro. And the route to market, just in terms of, this is a bit outside my area of expertise. So how will you take this device to market? Will you find partnerships? Will you license it? Will you manufacture and sell? How do you envisage this?
So we will consider partnerships, but right now the business plan would be to do it as a service first. So pharmaceutical companies and research centers will send their cells to us. We would sort, isolate, and characterize the cells based on their ability, and then send it back to the pharmacy.
Have you spoken to some of the pharmaceutical companies?
Yes.
To gauge interest?
Yes. And they like it because a lot of the current technology is too expensive and inaccessible. So they like it, but they would need a bit more results in the in vivo test.
And would those, any of the work you'd be doing, would they be partitioned? So you would only be able to work in certain areas, certain fields with particular pharmaceutical companies, or would you try to be less exclusive in any of your partnerships?
We are not sure. That will be done in the business plan later on. Yes.
And how much do you think pharmaceutical companies, or how much do you think you can charge pharmaceutical companies for this screening service? What's it worth to them?
So each service could cost around $80,000 to $100,000 for each cells population of cells that they.
Well done. Thank you very much. So